continue with our discussions, we go now to secondary structure of protein. That is the next higher level of macromolecular structures. When you say macromolecular structures, these are big structures of molecules. So the term is macromolecules, and they are known to be biomolecules also. So Skanda structure, it has a regular special arrangement of atoms along the chain backbone. So later I can show you about the backbone, wherein there's spatial, when say spatial arrangement, there's a space between the two chain of a polypeptide. So it is stabilized by weak non-covalent interactions. There is no sharing of electrons, and such as hydrogen bonding or HH bond. So there is what we call hydrophobic bonding. So when you say hydrophobic, it is considered to be reactions where it repel water, and electrostatic attractions, meaning there is an attractions between two opposite ions, the positive and negative charged ions, that is found in the second structure of the proteins. So to differentiate, so we have the primary structures, this one. So we have the amino, the terminal end, and then another terminal end, the carboxyl end. While secondary structure is composed of two amino acids, wherein hydrogen bonds between amino acids at different locations of the polypeptide chain. So you try to say there is a bonding of hydrogen ion, okay, to the carboxyl ion. Okay, there are two types of this one: secondary alpha and the beta, or the pleated sheet. This is alpha helix and this is pleated sheet. So try to see there's a bonding between the hydrogen and oxygen bond. This is considered to be electrostatic bonding because of the two opposite charges of ions that they bond together. Okay. Now, tertiary structure, we have beta polypeptide. The hemi group is found in example the hemoglobin. Okay, and we have quaternary. So we try to see the hemi. There are at least four hemi group that are bound to each other. Okay. Two principal secondary structures are alpha helix and beta helix. So when you say alpha helix, the telephone cord shape, okay, held by hydrogen bonding or hydrogen-nitrogen bonding, and a group of carbon to oxygen carboxyl group. So this is bonding between the hydrogen and the carboxyl group. Okay, group next turn of helix for 4 amino acids down the chain. So the typical alpha helix is almost 11 amino acids long, while beta sheet, or the pleated sheet structure of beta sheet, is held together by hydrogen bond between amide groups of the linear and average number of amino acids. Typically, beta sheet is 6 average of 6 strands bonded together. So strands meaning it is the what we call this one, the chain of amino acid, that is meaning the strand. Okay, next, so secondary structure, this is alpha helix, so bonded by hydrogen. We have the beta pleated or beta sheet, beta strand shown in flat arrow pointing to the carboxyl group. This is hydrogen bond. Next, secondary protein structure, this one is the alpha helix. So this is connected by a loop. So later we'll discuss about the loop. Try to see the beta sheet. If we have this one, this is pleated, okay, and this is the telephone cord, or the alpha helix, or the alpha helix. So there is a term as beta turn. The high proportion residues is tightly folded by proteins involved in turns of backbone. So that is considered turning. So in the backbone, if you try to see, this is the parallel or top view of the beta sheet, and we have the side view, and these turns may be flexible loops, amino acids serving as different stretches of secondary structure. They may be sharps, turns reverse to the chain direction. So every example, this is the beta sheet, so it is protected by a loop or flexible loop. So flexible loop helps in the reversal of the amino acid, because in the inside that DNA, for example, or inside one protein, okay, inside, that is a part of the protein wherein proteins are considered to be mobile. So there is what we call tendency of the reversion or the reverse of the chain, because it is considered to be the movement of the protein. Based on the movement of protein, so there is a reversal, there is a reversal of the structure of the protein, especially in the beta sheet. So it is protected by a flexible loop. One of the structural element connections, anti-parallel beta sheet structure in beta turn. So this is considered to be a beta, and when you say anti-parallel, there is what we call an opposite directions, so or opposite arrangement of a beta sheet. So meaning that is anti-parallel beta sheet. Try to see this one, anti-parallel. So the arrow or the position, the structure of the beta sheet are considered to be opposite. Next, beta turn usually present in proline and glycine. If this is the pleated or beta sheet, so this is considered to be the beta turn. Where in beta turn, it is protected by a flexible loop.